Hello, everyone. My name is Nicholas Jajos, and I'm really excited to be here to discuss my study on identifying genetic biomarkers for essential tremor diagnosis. I chose this topic because I've seen essential tremor all my life. My father has essential tremor, and it hurts seeing him struggle with trying to hold a fork or write. And I wanted to get to the bottom of this neurological disease. To begin, essential tremor affects 7 million Americans, or 4% of the global population of adults 40 and up. It's characterized by involuntary and rhythmic shaking of the hands, and it affects the cerebellum, the motor control section of the brain, specifically Purkinje cells, which are these cells that exhibit inhibitory signals to other neurons. ET has been associated with increased risk of life-threatening neurological diseases like dementia, Alzheimer's, and Parkinson's disease. With that said, there's been a relative lack of progress in understanding ET's genetic makeup, namely because of the lack of an official clinical diagnosis. With that said, technological advancement in RNA sequencing and this new wealth of bioinformatic data available has made it easier to analyze diseases like ET. Yet these underlying genetic pathways of ET still remain elusive, and very few studies have combined genetic information and machine learning to diagnose ET. The objectives of my study are to 1. Identify genetic pathways associated with ET, 2. Build machine learning models to differentiate between ET and control, and 3. Find and elucidate the most important genes with machine learning between ET and control for drug treatment. I started with attaining a data set of raw RNA sequence data from NCBI-GEA, and I had 55 samples taken from the postmortem cerebellum. I performed differential gene expression on this data set, and then using those genes found, I conducted gene set enrichment analysis. I then ran random forest and logistic regression models for feature importance and gene identification. Here are my results. I ran DC2, which is this RStudio package for differential gene expression, and I found 86 significant genes with a p-value of less than 0.01 and an adjusted p-value or FDR of less than 0.25 from a set of 23,000 genes. Now, the FDR value of less than 0.25 is not arbitrary. It's been used in similar studies before. And on the right here are the top 10 significant genes from my DC2 analysis, with notably a negative log fold change indicating downregulation and a positive one indicating upregulation. Getting these 86 genes is really important because I need to find these 86 genes to put into my other models in order for the information to be accurate. If I used all 23,000, then there would have been too much noise in all of my models. I then ran gene set enrichment analysis, specifically with the hallmark gene set, and I found 20 unique pathways. Now, association is not causation. So I looked into these pathways further, and I found specifically that the fatty acid metabolism pathway is associated with Alzheimer's disease, and the cholesterol homeostasis pathway being downregulated is also associated with other neurodegenerative diseases. It's worth to note that these pathways have not been associated with ET before, so this is a novel finding. I also used gene set enrichment analysis with KEGG, which is similar to the hallmark gene set, and I found 32 unique pathways. Specifically, I found that the KEGG ribosome pathway was downregulated, and this could be possibly stalling the Purkinje cell development found in the cerebellum. Specifically, ribosomes are these protein factories, and if these aren't being produced at a steady rate, then cell death will occur. I also found that axon guidance was upregulated, and axons are basically carry these nerve impulses away from nerves. So if these are being upregulated or overstimulated, it could be resulting in that characteristic tremor. And this axon guidance being upregulated has been corroborated with other studies before. I also found that Parkinson's disease was being downregulated in this essential tremor versus control. And this could indicate the increased death of dopaminergic neurons, either in the cerebellum or perhaps even in the substantia nigra. adding to the fact that ET could be a precursor to Parkinson's. And the fatty acid metabolism pathway, though not meeting the requirements of P less than 0.01, was also downregulated, and it matches the hallmark set. So this is worth noting because it's both in the hallmark and in this KEGG gene set. I then ran random forest, which is a binary classifier that differentiates between ET and control by generating thousands of these decision trees and taking the majority. On the right here is a sample of one of the decision trees used for my first run with random forest, where I averaged 80% accuracy after inputting 86 genes, the same 86 genes from differential gene expression. I then narrowed those 86 genes down to 32 genes using feature optimization, and I increased the accuracy of my model by 5%, up to 85%. I then ran logistic regression, which is another binary classifier model, using, again, the 86 genes from differential gene expression, and I had a 78% accuracy. I then ran this logistic regression algorithm on each of the random forest special feature optimization genes, and I found 8 genes by themselves had 80% or higher test accuracy. This means that, for example, the SF2PA2 gene, just by looking at the raw RNA sequence, my model was able to differentiate between ET and control 93% of the time. And these top 8 genes are really important because by identifying these genes, gene therapies or drug development can be used to better solve ET. All in all, I found 8 genes with large logistic regression importance, and this is going to result in targeted drug treatment for ET and a better understanding of this really understudied disease. It contributes to scientific knowledge with fatty acid, cholesterol, ribosome, axon, and Parkinson pathways now being associated between ET and control, which haven't been seen in other studies. And, finding these pathways, I found these pathways in other neurological disorders like dementia and Alzheimer's, indicating that ET could also be related to these neurological disorders. I also have clinical machine learning significance, as my model can be used to predict ET off of raw RNA sequence data from brain biopsies. As far as future investigations, to really know if these genes are the genes causing ET, I could target these genes in a wet lab in model organisms, and I could also look further into the specific genetic pathways of these 8 genes. I could also utilize machine learning models with higher statistical power like neural networks. All in all, my study is important because I identified 8 genes between ET and control that can be used for drug development, as well as 5 pathways that could be used to understand ET further. And here are my references. Thank you all for listening.